There is a bottle of it in your house right now. You have taken it. Your parents gave it to you when you were small. Paracetamol. Acetaminophen, if you grew up in America. The most widely used painkiller on the planet. Doctors have handed it out for 70 years. It is on the World Health Organization's list of essential medicines. But after all that time, science still cannot fully explain how it takes your pain away. We can describe exactly how it destroys a liver, enzyme by enzyme. So why can nobody agree on how it stops a headache? 1877. Baltimore. A chemist makes something he never uses. This is paracetamol. One benzene ring, a hydroxyl group, an amide tail. A small, unremarkable molecule that would go on to reach more human beings than almost any other drug ever made. In 1877, a chemist named Harmon Northrop Morse reduced p-nitrophenol with tin in glacial acetic acid. He was working at Johns Hopkins University in Baltimore. He became the first person to hold paracetamol, and he never gave it to a single patient. It sat in the chemical literature for 16 years before anyone tried it on a person. To see why nobody cared, you have to remember what a fever meant in the 1880s. Doctors could measure it and chart it. Bringing it down reliably was another matter entirely.
In normal human terms, this is a family tree. Coal tar fed the German dye works. The dye works made aniline, and aniline made a shelf of chemical cousins, acetanilide, phenacetin, and paracetamol, 3 relatives that all break a fever, and only one of them is still legal. The first of those cousins arrived in a Strasbourg hospital in 1886, and the story of how is famous. The version everyone tells goes like this. 2 young assistants of Adolf Kussmaul were treating a patient for intestinal worms. One of them was Arnold Kahn. The pharmacy sent up the wrong jar, and what arrived was acetanilide. The worms were unaffected. The patient's fever, however, dropped away within hours. Kahn and his colleague Paul Hepp published that same year under the title Antifebrin, a new fever remedy. Within months, the drug was being sold across Europe as a cheap rival to everything else on the shelf. One accident, one paper, and an entire class of medicine was born. Acetanilide reached the market in 1886 under the name Antifebrin. But here is the problem with that story. Kahn and Hepp's own paper never mentions worms, or naphthalene, or a mix-up at the pharmacy. It says something much plainer. What they wrote was that a fortunate accident had placed the preparation in their hands. That is the whole account. The worms and the wrong jar were added by people retelling it later, and the retelling has outlived the paper. A good origin story is hard to kill, even in medicine. What is not in dispute is what happened next. Doctors started reporting that patients on acetanilide were turning a dusky blue. The drug was damaging hemoglobin, the protein that carries oxygen, until the blood could no longer deliver it. So the industry had a drug that broke fevers and a drug that starved blood. They were the same drug. The company that solved it was Bayer, and the reason is almost comic. Bayer had a mountain of an industrial byproduct called 4-nitrophenol, left over from manufacturing a blue dye, and it badly wanted something to do with the stuff. It was industrial waste that Bayer needed to turn into something worth selling. A researcher named Oscar Hinsberg took that waste and ran it through 3 chemical steps. What came out the other end was phenacetin, a well-tolerated painkiller that brought down a fever without turning anybody blue. By 1887, both drugs were on sale, antifebrin and phenacetin, born out of the same dye works. The race was on to find a cousin that kept the fever and lost the poison. The winner would be a molecule called phenacetin. 1893. One man tests it and gets it wrong. In 1893, a clinical pharmacologist named Joseph von Mering became the first person to give paracetamol to human beings. He was not a minor figure. He was one of the most respected experimental physicians in Germany. And his verdict on the drug was simple: it was dangerous. Von Mering reported that paracetamol did the same thing acetanilide did. It turned the blood, he said, in the same dangerous way. On that basis, the molecule was set aside. He was wrong. That single verdict cost the world 54 years, from 1893 until 1947, and it handed the market to a cousin that would eventually be banned. That is not a rounding error in medical history. That is 2 generations. The cousin was phenacetin, and for half a century it was everywhere. Phenacetin went into headache powders, cold remedies, and mixed analgesic tablets sold in every pharmacy in Europe and North America. It had been introduced in 1887, and it became one of the most heavily consumed drugs of the 20th century. Look at what was in one of these powders: phenacetin for the pain, caffeine to lift you, aspirin alongside it. People took them daily, for years, and nobody connected the powders to what was happening in their kidneys. By 1950, doctors were beginning to describe a pattern of kidney damage in people who used analgesics long-term. It took decades to name it, and decades more to act on it. In the Swedish factory town of Huskvarna, workers found that the powders made them faster, stronger, and less tired. Piece rates meant that mattered. At the peak in the 1950s, one drugstore in that town was selling 8,000 sachets a day. Apprentices were handed them as a tip for running errands. The same thing was happening in the Swiss watch industry, where phenacetin powders were freely available in bulk to the workforce. Then came localized epidemics of kidney failure in Sweden and in the watch towns. In the mid-50s, a company doctor finally worked out what the powder was doing to them. 8,000 sachets a day, from one shop, in one small town. That is what an unregulated painkiller looks like when it also happens to be habit-forming. Keep phenacetin in mind. The drug that beat paracetamol in 1893 is the one that ends up banned, and that comes later. The difference between them is almost nothing. Swap one group of atoms and phenacetin becomes paracetamol. Your liver does that swap for you. Meanwhile, the molecule Morse had made in Baltimore sat unused. Nobody sold it, nobody prescribed it, and it existed only as a line in the chemical literature. It had been condemned on a single verdict that nobody had gone back to check. That somebody arrived in 1947 in a laboratory working on a completely different question. 1947. Somebody finally checks the work. In 1947, 2 researchers named Greenberg and Lester worked out where acetanilide actually goes once it is inside you. It breaks down, and the thing it breaks down into was paracetamol, which meant that everyone who had ever taken antifebrin had, in effect, been taking paracetamol. A year later, 2 pharmacologists picked the work up and pushed it further. One of them was Bernard Brodie, a specialist in what the body does to a drug. The other was Julius Axelrod, then a young researcher at the start of a long career. Together with Frederick Flynn, they published 3 papers in the September 1948 issue of the Journal of Pharmacology and Experimental Therapeutics. Their finding was the one that mattered. Paracetamol is the active metabolite of both acetanilide and phenacetin. Every fever those drugs had broken since 1886 had been broken by paracetamol. The drug had been hiding inside the drugs the whole time. In 1955, McNeil Laboratories put it on sale in the United States as a liquid for children under a name you already know, Tylenol Children's Elixir. A year later, it reached Britain as Panadol, sold by Frederick Stearns and Company, on prescription only. and advertised as gentle to the stomach because the alternative was aspirin, a drug that chews at the stomach lining. By 1963, it was in the British Pharmacopoeia, and the climb was underway. A children's version, Panadol Elixir, followed in 1958, and the pitch never really changed. Fewer side effects than aspirin, little interaction with other drugs, safe enough to give a child. That reputation is what built the market. Safe enough to give a child. Hold on to that phrase. Over the following decades, it became the default, the thing you reach for before you consider anything else. It went into cold remedies, flu sachets, and migraine formulas, until it was hard to find a medicine cabinet without it. And every one of those boxes contained the same molecule, which is a detail that matters later. If you are finding this interesting, subscribing genuinely helps, because from here, it stops being history. So how does it actually work? Nobody fully knows. Here is the strange part. You take 2 tablets, and about half an hour later, the headache loosens its grip. That happens billions of times a year, reliably enough that we build health systems around it. Ask a pharmacologist to explain why, and the answer gets uncomfortable. Compare it to the drug beside it on the shelf. We know how aspirin works, and we know how ibuprofen works. They block enzymes called cyclooxygenase, which build the molecules that make injured tissue swell and hurt. The chain runs like this. Tissue gets damaged. Cyclooxygenase enzymes switch on. They build prostaglandins, and prostaglandins are what turn an injury into swelling and pain. Block the enzyme, and the pain signal never gets built. That model is clean, it is teachable, and paracetamol does not fit it. If paracetamol worked the way aspirin works, it would bring down swelling too. It barely does. Take it for a sprained ankle, and the ankle stays just as swollen, while the pain still fades. Ibuprofen brings the swelling down as well as the pain. Paracetamol mostly does not, which turns out to be a real clue. So for decades, the honest position in pharmacology was a shrug, dressed up in careful language. A recent review of the evidence put it about as plainly as a scientific paper ever puts anything, which is to say, cautiously. For a while, it looked like there was an answer. In 2002, a team reported a third cyclooxygenase enzyme, COX-3, concentrated in the brain and sensitive to paracetamol. It was tidy, it explained the central action, and textbooks started printing it. Then other laboratories went looking for COX-3 in human tissue. What they found did not behave the way the hypothesis needed. In humans, the variant produced a protein with a completely different amino acid sequence, one that did not do the job and was not inhibited by paracetamol. The tidy answer fell apart, which is how a mechanism can appear in a textbook and then quietly leave it again. The current thinking is stranger than the textbook version. Paracetamol may not be the active drug at all. Once inside you, part of it is converted into a second compound, and that compound is called AM404. And AM404 does not behave like a painkiller at all. It behaves like something the brain already uses. AM404 turns up in the central nervous system, where it activates a receptor called TRPV1, the same channel that responds to chili heat, and feeds into the cannabinoid system, the one your body uses to damp pain down on its own. So the picture inverts. Rather than blocking the signal at the injury, the drug appears to reach into the brainstem and lean on the machinery that already suppresses pain from above. You are not silencing the alarm, you are helping the building turn it down. And even that is not settled science. The picture is still moving. In 2025, researchers reported that AM404 is also made out at the nerve endings themselves, where it shuts down the sodium channels that carry pain signals. which would mean the drug works centrally and peripherally by two different routes. 150 years in, the mechanism of the most common painkiller on Earth is still an open research question. Not knowing the mechanism is not just an academic embarrassment. If you cannot say precisely how a drug works, you cannot reason confidently about who it will fail, what it will clash with, or how you would go about building a better version of it. 70 years of daily use, and the design notes for it are still missing. The honest summary is that paracetamol works reliably, that several mechanisms are probably running at once, and that the field has not finished deciding which of them matters most. Which would be a charming piece of trivia, except for one thing. There is a part of this drug's behavior that is not mysterious at all. We have mapped it in detail, enzyme by enzyme, because we had to. That part happens in the liver, and it is the reason this story has a body count. The part we understand best is the part that kills people. Take a normal dose, and almost none of this matters. Your liver grabs the drug, bolts a couple of chemical handles onto it to make it water-soluble, and your kidneys flush it out. That is the fate of the overwhelming majority of every tablet you have ever taken. But the dose splits three ways. Two of those routes are harmless housekeeping. The third is not. A small fraction gets picked up by an enzyme called CYP2E1 and turned into a compound called NAPQI, which is about as reactive and unwelcome as anything your liver produces on purpose. At a normal dose, that fraction is dealt with before it can touch anything. The thing that deals with it is glutathione, a molecule your liver keeps in stock for exactly this kind of problem. In normal human terms, it is a mop. NAPQI appears, glutathione binds it, and the whole mess leaves in your urine. The catch is that you only have so much mop. Push the dose up, and the two safe routes saturate. More of the drug gets shunted down the CYP2E1 path, more NAPQI is produced, and the glutathione supply starts draining. When it runs out, NAPQI has nothing left to bind except your own cells. Same molecule, same liver, same enzymes. The only variable that changed is how much of it arrived at once. What happens next is not subtle, and it is not slow. NAPQI binds to proteins inside the mitochondria, the parts of the cell that make its energy. Respiration falters, energy production stops, and liver cells begin to die in large numbers. That is acute liver failure, and it can follow a single afternoon's decision. In the United States, this drug is the single most common cause of acute liver failure. It accounts for roughly half of all reported cases and around a fifth of the country's liver transplant cases. Acute liver failure is not a warning sign. It is an organ shutting down. Once it develops, more than a quarter of those patients die, and about a third of them need a transplant to survive at all. And the person it happens to is often somebody who was treating a headache. The yearly figures are worse than most people would guess. Around 56,000 emergency department visits a year, roughly 2,600 hospitalizations, about 500 deaths, and more calls to poison control centers than any other substance on the list. And here is the part that reframes all of it. Around half of those poisonings were never intended. This is how it happens. The flu sachet has paracetamol in it. So does the night capsule. So does the painkiller in the drawer. Every label followed, the limit passed, and nobody in that story did anything reckless or even unusual. The margin is also not identical for everyone. Chronic heavy drinking raises CYP2E1 and lowers your glutathione reserve. So does prolonged fasting. The evidence does not say a normal dose becomes dangerous, but it does say repeated doses above the limit get riskier. Which brings us to the most dangerous version of all of this. It is not the single dramatic overdose, it is the staggered one. A few extra tablets tonight, a few more tomorrow, none of them anywhere near a headline number, quietly outpacing the liver's ability to keep up. And because the early symptoms are mild, people often arrive at hospital after the window has closed. There is an antidote, and it is a good one. In 1977, Prescott and colleagues published the treatment of paracetamol poisoning with N-acetylcysteine in The Lancet. It works by restocking the very thing the overdose drained. It gives the liver its glutathione back. But the antidote comes with a clock attached. Given within about 8 hours of the overdose, N-acetylcysteine is almost completely protective against death. Leave it later and the protection falls away, which is the cruel detail here, because in the first hours, a paracetamol overdose often feels like almost nothing at all. So the question becomes what you actually do about a drug like this. And the answer that worked best was not a chemical one. The fix, when it came, was not chemistry. First, remember phenacetin. The cousin that beat paracetamol kept its market for most of a century, and then the bill arrived. It was tied to kidney damage, and then to cancers of the renal pelvis and bladder. One country at a time, it was taken off the shelf. Canada withdrew it in 1978, Britain followed in 1980, and in November 1983, the American regulator ordered drugs containing phenacetin off the market entirely. The molecule that had been chosen over paracetamol was now the one nobody could sell. There is one more packaging story in this drug's history, and it is darker. In the autumn of 1982, 7 people in the Chicago area died after taking Tylenol capsules that somebody had opened and filled with cyanide. The case has never been solved, and nobody was ever charged. The response reshaped an entire industry. Congress made product tampering a federal crime, and regulators required medicines to be packaged so that interference would be obvious. Blister packs, foil seals, breakable caps. That is where all of it comes from. Twice now, the thing that made this drug safer was the container it came in, which left paracetamol standing alone, holding the entire market it had been denied for 50 years. And that created the problem this whole story has been walking toward. A drug in every home, sold in any quantity, with a gap between the helpful dose and the harmful one that is narrower than almost anything else in the cupboard. Britain decided to try something that sounds almost too simple to work. In September 1998, new legislation capped how many tablets you could buy at once. 32 in a pharmacy, 16 anywhere else. That was the entire intervention. It needed no new chemistry and no new antidote, just a smaller box. The reasoning was that a large share of these overdoses are impulsive, taken with whatever happens to be in the house. Shrink what is in the house, and you put a small amount of friction between an impulse and a lethal quantity. Researchers then spent 11 years measuring whether it had done anything at all. Deaths from paracetamol poisoning in England and Wales fell by 43%, that is, 765 people. Liver transplants for paracetamol damage fell by 61%, from a change to the size of a box. Britain was not the only place that tried shrinking the dose rather than fixing the drug. In January 2011, the American regulator told manufacturers that combination products mixing paracetamol with an opioid could carry no more than 325 milligrams of it per tablet. Some formulations had been carrying more than twice that. Companies had until 2014 to comply, and researchers watched what happened to the numbers for the next 8 years. Hospitalizations for paracetamol and opioid toxicity fell from 12.2 per 100,000 to 4.4. And among acute liver failure cases, the share involving these combination products dropped from 27% to 5%. Meanwhile, cases involving paracetamol on its own went up, which is how you know the rule is what did it. A century of chemistry could not make this molecule safe. A rule about packet sizes measurably did, which is a strange lesson for a story that began in a dye works. Paracetamol was synthesized by a man who never used it, condemned by an expert who was wrong, ignored for 50 years, and rescued by accident. We still cannot fully explain the good it does, while the harm has been mapped enzyme by enzyme. And it is still the drug we reach for first, because on the whole, and at the right dose, it works. There is a bottle of it in your house right now. You know more about what is inside it than most of the doctors who prescribed it for the first 100 years. If you want the other half of this story, phenacetin is the one that won that argument and spent 90 years damaging kidneys before it was banned. That video is next. Tell me in the comments whether you ever checked your cold and flu sachet.